Chapter 4: A Guided Tour Through Your Brain. You, your joys and your sorrows, your memories and your ambitions, your sense of personal identity and free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules. Sir Francis Crick, The Astonishing Hypothesis. Why do you eat? You're bored. You have a craving. The clock says it's time to eat. It's a special occasion. Your grandmother made your favorite dessert. Your brain and the rest of your body needs nutrients. It can be easy to forget this last one, but it's the original and only reason why all living creatures must eat. To build and maintain your cells, you need raw materials, parts, and a fuel source, energy. And these come from macronutrients, micronutrients, oxygen, sunlight, and water. Macronutrients are versatile molecules we use in large amounts: protein, fat, cholesterol, and carbohydrate. All of these can be used as building blocks to construct vital brain components like membranes that surround every cell and keep its contents organized. Macronutrients can also be burned for energy. Micronutrients are substances we use in small amounts to help build, maintain, and energize the brain. These include vitamins, complex molecules, minerals, simple salts, and essential fatty acids. Like a construction site without workers or tools, without micronutrients, all you'd have is a pile of construction materials and an energy source with no way to use either one. In our quest to understand which foods best nourish, protect, and energize the brain, let's embark on a tour of the brain's interior to learn what it's made of and how it works. Neuroscience is complex and can feel overwhelming, but understanding the basics might just inspire a sense of wonder about what your brain cells do all day long and help motivate you to take good care of them. It will also give you the foundation for understanding some of the things that can go wrong inside your brain and how changing your diet could help. As we explore the fascinating inner world of our most prized organ, keep in mind that absolutely everything we'll encounter along the way comes from food, water, air, and sunlight. The cloistered brain. Hidden inside a fortress of bone about 1/4 of an inch thick, the brain is the most heavily guarded organ we possess. Fragile and gelatinous, this collection of billions of cells floats in an 8-ounce tank of nourishing cerebrospinal fluid that makes the 3-pound brain feel as if it weighs only 2 ounces. Additional layers of security are provided by 3 protective membranes called meninges that surround the brain, tether it in place, and cushion it against traumatic injury. Yet the brain's most advanced defense system is invisible to the naked eye. The microscopic capillaries that crisscross the brain, delivering oxygen and nutrients to its hardworking cells, are uniquely designed to prevent risky molecules, toxins, and infectious organisms from escaping the bloodstream and invading the brain's sensitive interior. Elsewhere in the body, these minute blood vessels are intentionally leaky to allow easy exchange of nutrients and waste products between the bloodstream and surrounding tissues. But the endothelial cells that form the walls of brain capillaries are joined together by interlocking tight junctions, creating a leak-proof seal. This blood-brain barrier is outfitted with intelligent pumps and transport channels that allow it to carefully control which molecules enter and exit the brain's sensitive interior environment. Situated all along the blood-brain barrier are transport proteins and receptors designated to escort essential macronutrients and micronutrients into the brain. A most peculiar place. Before we explore the inner workings of the brain, let's first stand back to appreciate its unusual exterior architecture. The outermost layer of the brain consists of a gray sheet of tissue about 1/10 of an inch thick, called the cerebral cortex, Latin for brain bark. Packed with layers of busy brain cells, the cortex is responsible for our most sophisticated intellectual functions, such as language, reasoning, and planning. The more intelligent a creature is, the larger the surface area of its cerebral cortex must be, which explains the human brain's strange topography. Its crumpled landscape of prominent folds and deep ravines was evolution's clever way of fitting the vast expanse of our cerebral cortex inside the skull. Just below this veneer of gray matter lies a thick zone of white matter, brain cell extensions wrapped in thick layers of myelin, an insulating material rich in fat and cholesterol. These cables of white matter connect hubs of gray matter with each other, forming an electrical grid that allows coordinated communication across the entire brain. Connecting the brain to the spinal cord is the brainstem, an evolutionarily ancient region responsible for controlling basic functions such as breathing and pumping blood. Protruding conspicuously from the rear of the brain is the cerebellum, Latin for little brain, which coordinates movement and balance. Within the brain's core lie dense collections of cells that work together to perform specialized tasks. Examples include the hippocampus, a seahorse-shaped region where learning and memory take place. This part of the brain is one of the first to malfunction in Alzheimer's disease. And the hypothalamus, which releases hormones involved in regulating appetite, metabolism, and reproductive cycles. Deep within the brain lie curiously shaped reservoirs called ventricles, full of cerebrospinal fluid. Cerebrospinal fluid circulates throughout the central nervous system, bathing the brain and the entire length of the spinal cord. This is the same fluid collected during a lumbar puncture or spinal tap. This solution, which replenishes itself roughly 5 times per day, helps deliver glucose and minerals, such as sodium, potassium, and magnesium to the brain, and flushes the brain of toxic waste products and defective molecules that would otherwise accumulate and harm brain cells. Your brain washes itself most effectively overnight during periods of REM sleep, rapid eye movement sleep, when you are dreaming. Just one of the many reasons sleep is vital to brain health. Let's now turn our attention to the approximately 170 billion individual cells that make up the brain. Only about half of these are neurons, the brain cells famous for conducting electricity. The other half is a diverse collection of glial cells, or simply glia, Greek for glue, which protect and serve neurons. Neurons, engineered to conduct electricity. Your brain's neurons must be in constant communication with each other and with the rest of your body's nervous system to smoothly coordinate all of your thoughts, feelings, and behaviors into the cohesive whole that is you. This impressive operating system requires 2 things: speed and specificity. Speed, so you can think, move, and react quickly. If it takes too long for you to reflexively pull away when you accidentally touch a hot stove, you could suffer serious burns. Specificity, so you can think, move, and react appropriately. After all, how long would you last if you responded to the sensation of touching a hot stove by moving closer to it? For speed, neurons are engineered to conduct electricity, which allows them to send messages at velocities as high as 200 miles per hour or more. For specificity, neurons produce a variety of neurotransmitters, such as serotonin and dopamine, capable of delivering different instructions depending on their type, location, and circumstances. The receiving ends of neurons feature extensions called dendrites, studded with dendritic spines, tiny docking stations where neurotransmitters from surrounding neurons briefly make contact to drop off their messages. Although a typical neuron only has about 6 dendrites, it may sport up to 200,000 dendritic spines, allowing it to listen to thousands of neighboring neurons. Depending on the amount and type of neurotransmitter, the receiving neuron may be stimulated enough to fire a jolt of electricity through its cell body and along its axon to the axon terminals at its tip, where fresh packets of its own neurotransmitters are stored at the ready. When the wave of electrical energy reaches these packets, they break open, releasing neurotransmitters into the gap that separates neurons from each other. These intimate spaces between the axon terminal transmitters of one neuron and the dendrite receivers of the next neuron are called synapses. You will find a diagram of the anatomy of a neuron in the accompanying PDF. Neurotransmitters attach only momentarily to receptors on the dendrites of nearby cells to deliver their messages. Then, as soon as they've accomplished their mission, their parent neuron vacuums them back up, recycling and repackaging them into fresh new storage sacks to wait for the next wave of electricity to set them loose again. Neurotransmitters. The brain uses dozens of different neurotransmitters. Those most familiar and directly relevant to mental health are serotonin, dopamine, norepinephrine, acetylcholine, glutamate, GABA, and melatonin. A table describing each neurotransmitter's role in the brain and examples of common medications designed to influence each neurotransmitter can be found in the accompanying PDF. How psychiatric medications work. Most psychiatric medications are designed to stimulate or suppress the activity of neurotransmitters. A familiar example is the antidepressant family of drugs called serotonin reuptake inhibitors, or SRIs, formerly called SSRIs, or selective serotonin reuptake inhibitors, such as fluoxetine, Prozac, sertraline, Zoloft, and escitalopram, Lexapro. One theory about the cause of depression is that serotonin activity is too low. As the name implies, serotonin reuptake inhibitors inhibit the reuptake of serotonin. In other words, they slow down serotonin recycling, which allows serotonin to spend more time in the synapse trying to communicate with other cells. While this strategy sounds logical, it focuses on only one small element of an intricately interconnected system, which may help to explain why medicines targeting serotonin don't always help as much as we wish they did. For serotonin, or any neurotransmitter to function properly, the whole neuron must be healthy, from the tips of its dendrites to the outer reaches of its axon terminals, and its surrounding environment and neighboring cells must be healthy as well. To fire or not to fire? If all of your brain's neurons were firing all the time, chaos would ensue. So we have elegant systems in place to carefully control their activity. When these regulatory systems malfunction, which is the case in bipolar disorder and in epilepsy, brain cells might fire when they're not supposed to, or they might not fire as often as they should. So, how does any given neuron in your brain know when to rest and when to fire? When a neuron releases a neurotransmitter into a synapse, that neurotransmitter then attaches to a receptor on the surface of a neighboring neuron's dendrite and sends a signal to that cell, either encouraging it to fire, excitation, or discouraging it from firing, inhibition, depending on the neurotransmitter and the receptor type. The dendrites on the receiving end of a neuron may contain a variety of receptors for a variety of neurotransmitters, but the axon terminals on the transmitting end of a neuron specialize in making, storing, and releasing only one neurotransmitter. It's as if each neuron can understand an entire vocabulary, but only knows how to say one particular word. This multiple input, single output system allows each neuron to weigh the opinions of thousands of its neighbors before deciding whether or not to fire. The electricity generated by a firing neuron is made possible by essential minerals that come from the foods you eat and the water you drink. Important examples include sodium, potassium, and calcium, which are all positively charged electrical particles. The chemistry term for these charged salt particles is ions, and the everyday clinical term for these is electrolytes. A neuron at rest is anything but relaxed. Thanks to hard-working sodium-potassium pumps embedded along the length of its axon's membrane, it's quietly brimming with electrical potential. Left to their own devices, sodium and potassium ions would simply distribute themselves uniformly within and around the axon until the concentration of sodium and potassium inside and outside the cell became equal. The pumps create an electrical imbalance between these two minerals by forcing lopsided exchanges of sodium for potassium. For every 3 sodium ions pumped out, only 2 potassium ions are pumped in, until the inside of the axon is negatively charged compared to the outside. Sodium and potassium would love to equalize their concentrations and relieve this electrical tension, but the only way they can cross the membrane is through special channels which are usually closed, trapping sodium and potassium in a high-energy state of imbalance. Like a slingshot stretched to its maximum length, the resting neuron sits primed and ready to fire at a moment's notice. Only if incoming neurotransmitter signals are strong enough to excite the cell to fire will sodium channels fly open and allow sodium ions to rush back into the cell. This wave of positive electrical charge created by incoming sodium particles zips all the way down the axon to its terminal tips, where it forces calcium channels open. Calcium then storms into the axon terminal, triggering neurotransmitter storage pouches to burst open and release their contents into the synapse to stimulate the next neuron in the chain. Diagrams of sodium-potassium pumps in a resting neuron and in a firing neuron can be found in the accompanying PDF. As you might imagine, this exquisite electrochemical cycle, which is taking place in tens of billions of neurons as often as 300 or more times per second, demands a tremendous amount of energy and a steady supply of nutrients, and the electricity it generates must be controlled to prevent the brain from short-circuiting. This is why behind every flashy electrical neuron, there are trusty glial cells. These underappreciated caretaker cells come in 4 varieties, each devoted to supporting high-maintenance neurons in its own way. Oligodendrocytes. These octopus-like cells sprout long membrane arms that wind themselves around the axons of nearby neurons, as many as 100 times, like a roll of tape, insulating them in a thick, fatty coating of myelin. A single oligodendrocyte can produce up to 80 myelin coils. It is myelin that makes white matter appear white, and that enables electrical signals to race swiftly and efficiently through the brain and the rest of the nervous system. The importance of myelin is made plain in multiple sclerosis, an autoimmune disease in which the body attacks and destroys myelin sheaths. Axons laid bare by multiple sclerosis are vulnerable to damage and can't conduct electricity properly. Brain symptoms of this devastating disease can include slurred speech, double vision, depression, and psychosis. A diagram of an oligodendrocyte can be found in the accompanying PDF. Astrocytes are star-shaped guardians of synapses. Synapses are where neurotransmitters are manufactured, so they have a voracious appetite for amino acids, vitamins, and minerals. Like attentive parents, astrocytes feed them, fuel them, and clean up after them, ensuring that these bustling cellular intersections always have everything they need in just the right amount. Microglia, the immune cells of the brain, are branched cells that vigilantly patrol their local neighborhood, keeping things tidy and looking for threats. If one of their many probing fingers encounters an invader such as a virus, the entire cell will instantly morph into a Pac-Man-like configuration, swallow the virus whole, and then revert to its original shape. Microglia play a major role in brain inflammation, one of the root causes of psychiatric conditions. Ependymal cells line the walls of ventricles, continuously producing fresh cerebrospinal fluid and encouraging it to circulate by waving long extensions of their bodies called cilia back and forth. Now that you've been introduced to the brain's outer contours and its microscopic structure, it's time to go down to the molecular level to discover which macronutrients and micronutrients you need in order to nourish, protect, and energize this magnificent organ. Your brain is made of macronutrients. Protein, fat, carbohydrate, and cholesterol are macronutrients, versatile molecules we use in large amounts, either as building blocks or fuel sources. All 4 macronutrients are made of carbon, hydrogen, and oxygen. But protein is unique in that it also contains nitrogen. The body can turn fat into carbohydrate or carbohydrate into fat, but it can't turn fat or carbohydrate into protein. This explains why you can survive eating a very low-fat diet or a very low-carbohydrate diet, but you cannot survive for long eating a low-protein diet. Protein, amino acids. Protein is the most important macronutrient in our diet. It is often said that we must eat protein regularly because it's the only macronutrient we can't make or store, but that's not quite right. While it's true we have no way to store it, we do make all of our own proteins out of amino acids. So we don't eat protein for the proteins themselves. What we're after is the amino acids they contain. Case in point, no matter how much calamari you consume, you will never be able to sprout a suction cup. Instead, you break the proteins in calamari down into individual amino acids and rearrange them into the human proteins you need to build a hair follicle, a hormone, or a hamstring. Just as we can create thousands of different English words using only 26 letters, we can build thousands of different proteins using only 22 amino acids. 9 of these amino acids are essential, impossible to make ourselves. 8 are conditionally essential. We can't always make enough to meet our needs. And the remaining 5 are non-essential. We make them ourselves. A table of these amino acids can be found in the accompanying PDF. We must eat all 9 essential amino acids regularly, and we must also eat the 8 conditionally essential amino acids when protein requirements are higher, including times of growth, stress, injury, illness, and during pregnancy and breastfeeding. Like all organs, the brain uses amino acids for a multitude of purposes. For example, most neurotransmitters are made from amino acids. The enzymes needed to build those neurotransmitters are also made of amino acids, as are the receptors that receive their messages. A diagram of how neurotransmitters are made can be found in the accompanying PDF. The nutrition-neurotransmitter connection. Neurons transform amino acids and other nutrients into neurotransmitters using chemical reactions that involve enzymes, vitamins, and minerals. So if you are short on certain nutrients, your neurons will not be able to manufacture all the neurotransmitters your brain needs to function properly. Notice, for example, iron and vitamin B6 are required to make the neurotransmitters serotonin and dopamine. If you have iron deficiency, you don't just have a blood problem, you have a brain problem. The ideal daily requirement for protein remains a topic of debate in nutrition science circles, with most sources recommending a minimum of between 0.4 to 0.6 grams per pound ideal body weight per day. I'll help you estimate your daily requirement in Chapter 17, but to give you a sense of how broad the range can be, if your ideal body weight is 140 pounds, many experts would recommend you eat at least 56 grams of protein per day, equivalent to about 10 ounces of salmon, while others would recommend at least 84 grams per day, equivalent to about 15 ounces of salmon. Amino acid deficiencies are unlikely, so long as you have access to adequate amounts of high-quality protein, meaning foods that contain a healthy balance of these amino acids. As we'll explore in more detail later, nearly all animal proteins contain the full complement of amino acids, whereas some plant proteins are low in certain amino acids, most commonly methionine, cysteine, and lysine. Fat, fatty acids. Just as protein molecules are made of individual amino acids, fat molecules are made of individual fatty acids, because each one contains 3 fatty acids attached to a simple sugar backbone called glycerol, like 3 flags flying on a flagpole. A diagram of a triglyceride molecule can be found in the accompanying PDF. Standard cholesterol blood tests include a test for triglycerides, which is a measure of how much fat is traveling through your bloodstream. Fatty acids come in a variety of lengths, short, medium, or long, and types, saturated, monounsaturated, or polyunsaturated. Saturated fatty acids are straight carbon chains saturated with the maximum possible number of hydrogen atoms attached to carbon atoms by strong single bonds. Saturated fats are very stable and tend to be solid at room temperature. Coconut oil and butter are familiar examples of fats high in saturated fatty acids. Monounsaturated fatty acids, or MUFAs, are bent in one spot where a fragile carbon-carbon double bond replaces strong carbon-hydrogen single bonds. This weak link causes MUFAs to melt more easily. MUFAs are liquid at room temperature, so we call them oils. Olive oil and avocado oil are high in MUFAs. Polyunsaturated fatty acids, or PUFAs, have even fewer hydrogens and contain multiple double bonds. Therefore, they contain multiple kinks. These oils are unstable and go rancid easily. Many nuts, seeds, and oily fish are naturally high in polyunsaturated fatty acids. Each of these fats plays important roles in our bodies, so it is incorrect to think of unsaturated fats as inherently healthy and saturated fats as inherently unhealthy. In fact, the majority of the fat in our bodies is saturated fat by design. Some of it is monounsaturated, but only a small percentage of it is supposed to be polyunsaturated. The liquid properties of unsaturated fats make them important ingredients in body fluids such as those that lubricate our eyes, tears, and joints, synovial fluid, whereas sturdier, denser saturated fats insulate us against the cold, cushion our internal organs against damage, and serve as a lightweight, compact, flexible way to store energy. Unlike plants, which store most of their energy as starch, think yams and turnips, we animals need to move about in the world and can't afford to be weighed down by unsightly potato-like structures. Fat also holds more than twice as much energy per gram as starch can, which is fortunate because hardworking muscles, hearts, and brains use a lot more energy than sedentary stems, leaves, and flowers. We have very limited capacity to store carbohydrate as starch, so the liver converts any surplus carbohydrate we eat into saturated fat for easy storage. This bears repeating. If you eat more carbohydrate than you can burn right away or store as starch, your liver will turn it into saturated fat, not unsaturated fat, because saturated fat is the most compact and practical way to store energy. It stands to reason that if saturated fat were inherently bad for us, the body wouldn't be designed to do this. If you were to try to store energy as unsaturated fat, you would sag everywhere and begin to resemble a Shar-Pei. Your brain is the fattiest organ in your body, other than fat tissue itself. If you could wring out all of its water, you'd be left with a wad of material containing about 50% fat. Why is the brain so fat? In a word, membranes. Membranes, which consist of 2 layers of fat, are flexible barriers that intelligently organize our bodies into individual compartments. Every one of your cells, and even many of the smaller structures inside your cells, are wrapped in membranes. Membranes contain a mixture of saturated fat for structural integrity and unsaturated fat for fluidity. The brain is particularly rich in fat compared to other organs, primarily because the myelin required to insulate brain circuitry is nothing more than tightly coiled membranes. And the brain's white matter contains more than 60,000 miles of myelinated axons. Roughly speaking, about 1/3 of brain fat is saturated fat, palmitic and stearic acid. And another 1/3 of it is a monounsaturated fat called oleic acid. Oleic acid is important to brain health, including for the creation of new memory circuits. But it is not an essential fatty acid, because brain cells can make it from other molecules. In fact, your brain is capable of building all of these fats from scratch out of simple glucose molecules. But it generally prefers to start with pre-assembled fatty acids, which it absorbs from the bloodstream. The remaining 1/3 of the fat in your brain consists primarily of two remarkable polyunsaturated fatty acids, an omega-6 fatty acid called arachidonic acid and an omega-3 fatty acid called docosahexaenoic acid, or DHA. Far from simple chains of stored energy, these elongated PUFAs possess unique properties that make them essential to the brain's specialized electrical functions. Arachidonic acid is a multi-talented molecule with 4 double bonds that force it into an unusual hairpin shape. It is required for brain development, membrane flexibility, cell signaling, and immune system function. DHA's 6 double bonds are arranged in a unique configuration that gifts it with quantum mechanical properties. More simply stated, DHA acts as a semiconductor of electricity. So we find it in electrical hotspots around the brain, synapses where electrochemical signals are being converted into memories, the retina of the eye, where sunlight is transformed into electricity, and the electron transport chain, where food molecules are turned into energy. More about this later. As distinguished Imperial College brain lipid scientist Professor Michael Crawford writes, DHA is essential for visual acuity and the truthful execution of the neural pathways which make up our recollections, information processing, and consciousness. DHA also directs the organization of the cerebral cortex, meaning that a deficiency of this precious fatty acid in early life could have profound and irreversible effects on many aspects of a child's intelligence, including critical reasoning skills, language, and learning capacity. One of the many services DHA and arachidonic acid perform in the brain is to generate first responder molecules for the brain's immune system. These fatty acids are firmly embedded within cell membranes, where they wait patiently until the immune system calls them up for duty. If the brain is under threat and needs to mount an inflammatory response, enzymes will free arachidonic acid from the membrane and convert it into a cascade of molecules that promote inflammation. When the time comes for healing, enzymes will free DHA from the membrane and convert it into a cascade of molecules that resolve inflammation. There is one more omega-3 PUFA that deserves special mention, even though the brain itself contains very little of it: eicosapentaenoic acid, or EPA. Just as DHA promotes healing in the brain, EPA promotes healing in the rest of the body. Our bodies can assemble most of the fats they need out of glucose or out of other individual fatty acids, but not EPA, DHA, and arachidonic acid. We don't possess the enzymes needed to make these special long-chain PUFAs from scratch. So we would be wise to eat foods that already contain them, and they are only found in animal foods. A far less reliable option would be to consume other long-chain PUFAs, namely linoleic acid and alpha-linolenic acid, both of which are found in plant fats as well as animal fats, and try to convert them into the PUFAs we need. But as we'll discuss later, not only is our capacity to do this extremely limited, but consuming more than very small amounts of linoleic acid is risky. Vitamins A, D, E, K1, and K2 are the fat-soluble vitamins, which means we can't absorb them unless we consume them with some fat. The standard recommendation is to obtain 20% to 35% of our daily calories from fat, and we are often advised to choose monounsaturated and polyunsaturated fats over saturated fats, a recommendation we will challenge throughout this audiobook. There is broad agreement that we require a source of essential PUFAs and that we must consume enough fat to absorb fat-soluble vitamins. Beyond these two undisputed recommendations, there is little agreement about the quantity and type of dietary fat humans should ideally eat. But as we'll see, there can be advantages to higher fat diets, particularly where the brain is concerned. Cholesterol. Most people view a high cholesterol meal as a heart attack on a plate, and most physicians think of cholesterol in the blood as something to be minimized with medication. But what exactly is it? Cholesterol is a hard, waxy substance essential for all animal life. We can't build hormones like estrogen, testosterone, or vitamin D without cholesterol, and its mandatory presence in our cell membranes should be comforting to us all, as its sturdy constitution contributes firmness and helps maintain their structural integrity. People often utter the words fat and cholesterol in the same breath, but not only do they look nothing alike, they serve very different purposes. You'll find an illustration of a saturated fatty acid and a cholesterol molecule in your accompanying PDF. Fat is a humdrum chain, whereas cholesterol sports a fancy honeycomb structure made of 3 hexagons plus a pentagon. In medical school, we affectionately called it 3 rooms and a bath. Our cells can make a triglyceride, fat molecule, in just 11 steps, whereas it takes more than 30 chemical reactions to hammer together a single cholesterol molecule. Saturated fat is easily chopped up and burned for energy, but cholesterol is indestructible. The only way to dispose of excess cholesterol is to send it to the liver to be turned into bile and excreted by the bowel. Although the brain represents only 2% of your total body weight, it contains 20% of your body's cholesterol, more cholesterol than any other organ in your body. What's all that cholesterol doing up there? Once again, the answer is membranes. A full 75% of your brain's cholesterol is wound up in myelin, and the rest resides in the membranes that line neurons and glial cells. In brain synapses, cholesterol plays an important role in cell-to-cell communication by helping to form lipid rafts, stiffened zones within cell membranes that stabilize the signaling proteins needed for neurotransmitter interactions. Cholesterol is too big and bulky to enter the brain across the blood-brain barrier, so not a single molecule of your brain's cholesterol comes from the cholesterol in your food. Instead, the brain makes all of its own cholesterol on site from scratch. I ask you, why would the brain go out of its way to intentionally produce large quantities of a substance that's bad for you? It wouldn't. It's really smart. It's a brain. Once the brain has gone to the trouble of building cholesterol, it tends to hold on to it for a long time. Cholesterol molecules inside the brain can last for years. And brain cells aren't the only cells capable of making their own cholesterol. In fact, every cell in the human body, except red blood cells, is equipped to do this. As for whether you can eat too much cholesterol, we'll explore that question in Chapter 11 when we discuss eggs. Carbohydrate. Much like fat, the sugars and starches that make up the carbohydrate family are simple, versatile molecules that can be easily chopped up and burned for energy, or combined with other molecules to build cell components. The 2 most important carbohydrates in our bodies are glucose, a simple sugar, and glycogen, a complex starch. Our bodies digest all of the sugars and starches we eat, whether they come from flour, fruit, or potatoes, down to glucose, a simple sugar ring that circulates in the bloodstream to supply cells throughout the body with energy. Certain organs, especially the liver and muscles, can store some glucose by linking tens of thousands of glucose molecules together into glycogen and squirreling it away for future use. You must have some glucose circulating in the blood at all times to feed your brain and other organs. The brain can only store a tiny quantity of glycogen. But that glucose does not need to come from your diet because your liver can make all the glucose your bloodstream needs out of fatty acids, and even certain amino acids if need be, via a process called gluconeogenesis, literally, new glucose formation. This means that eating carbohydrate is entirely optional. We'll cover carbohydrate in more detail in the coming chapters. I hope you've enjoyed our tour of brain anatomy. By seeing firsthand what the brain is made of, we can begin to assemble our brain-healthy diet ingredient list. The brain needs high-quality protein to provide it with the full complement of essential amino acids, such as tryptophan, which it uses to build serotonin, and tyrosine, which it uses to build dopamine. The brain makes its own cholesterol and can even make its own saturated fats and monounsaturated fats if need be, but it must have access to reliable sources of two polyunsaturated fatty acids, DHA, an omega-3 fatty acid, and arachidonic acid, an omega-6 fatty acid. The brain requires all essential micronutrients, with vitamin B1, vitamin B6, iron, choline, and copper being most directly involved in neurotransmitter production. Vitamins B9 and B12 support this process indirectly. And the brain requires electrolytes, mineral salts, to conduct electricity, with sodium, potassium, and chloride being most directly involved. For your brain to function at its best, it must be healthy through and through, which starts by nourishing it properly. If even one essential amino acid is in short supply, you won't have the ingredients required to build all of the vital molecules it needs, like neurotransmitters, receptors, or calcium channels. Without the right fats in the right proportions, brain development could go awry, membranes may become too flimsy or too rigid, and the brain's immune system could malfunction. Now that we know more about what it takes to nourish the brain's infrastructure, it's time to move on to the next pillar of robust mental health: brain energy production.